breastfeeding an opportunity to prevent malnutrition. Children are highly susceptible to growth faltering, especially between 6 and 24 months, when breast milk is replaced by low nutrient density foods. Additionally, it is very difficult to reverse stunting after the age of 2 years. Hence, complementary feeding period is considered a crucial window of opportunity to prevent undernutrition and its long-term negative outcomes. Introducing complementary foods too early, before 12 weeks of age, or too late, after 26 weeks of age, can exert undesirable health consequences. Early introduction may increase the risk of gastrointestinal and respiratory tract infections, obesity in later life, atopic eczema, and food allergy in high-risk populations. It may also increase weight gain, at least temporarily during infancy. Late introduction may increase the risk of nutritional insufficiency, immune system disorders, and development of type 1 or 2 diabetes in later life in high-risk populations. According to a study conducted in Bangladesh, exclusive breastfeeding beyond 1 year of age was associated with an increased risk of mortality in children aged 12 to 35 months, with the risk being higher among children aged 12 to 17 months. Breast milk provides all the energy and macronutrient needs up to 6 months of age. However, it does not provide micronutrients, such as iron and vitamin D, in sufficient quantities. Beyond this period, there is an energy gap of 200 kilocalories per day in infants aged between 6 and 8 months, 300 kilocalories per day in infants aged 9 to 11 months, and 550 kilocalories in children aged 12 to 23 months, which needs to be filled by complementary foods. Complementary foods should also meet the gaps in nutrients such as proteins, iron, and vitamin A. As can be seen in the graph, the largest gap is for iron. By 6 months of age, an infant's weight usually doubles and the infant also becomes more active. Exclusive breastfeeding is no longer sufficient during this period. Therefore, complementary foods should be introduced by 6 months of age to complement breastfeeding and to compensate for the nutrient and energy gaps. Infants may require iron supplementation if they reside in regions with anemia prevalence of greater than or equal to 40% in infants and young children. The 2008 statement by the American Academy of Pediatrics, AAP, recommends vitamin D supplementation in breastfed infants and in formula-fed infants if they are receiving less than 1 liter of formula. Further, fluoride supplements may be required for all children aged greater than 6 months if they live in communities with suboptimally fluoridated water, after carefully considering other fluoride exposures. Complementary feeding practices can be assessed at an individual level using a feeding history job aid. For children that are still breastfeeding, the feeding history job aid assesses how many times per day and night the infant is being breastfed, and if expressed breast milk is used, how is it stored? Information on other foods given, the number of meals and snacks per day, the quantity of food at each meal, consistency of the meal, inclusion of foods from animal sources, dairy products, dark green vegetables, fruits and pulses, assistance provided to the child during feeding, addition of vitamin or mineral supplements, and eating patterns during sickness are also included. Another method is the Infant and Child Feeding Practices Monitoring Tool and Guide, developed by the United States Agency for International Development. This tool helps to record practices related to 4 sets of indicators, namely breastfeeding. Now let's discuss the different guiding principles for complementary feeding. Infants should be exclusively breastfed from birth to 6 months of age. At 6 months of age, complementary food should be introduced while continuing breastfeeding on demand up to 2 years of age or beyond. Feed the child slowly and patiently. Encourage the child to eat and not feed forcibly. Assist older children when they are feeding themselves. Minimize distractions while feeding and talk to the child while feeding. maintaining affectionate eye-to-eye contact. If a child refuses to eat, try different combinations, tastes, and textures of food. Safe preparation and storage of complementary foods is to prevent microbial contamination of complementary foods. Therefore, instruct caregivers to avoid feeding liquids using bottles with nipples. Instead, cups and spoons should be used. Hands and utensils used for feeding should be thoroughly washed. Raw and cooked food should be separated. Food should be cooked thoroughly. Clean and safe food ingredients and water should be used, and food should be stored at safe temperatures. As the child grows, the intake of breast milk reduces and more food is needed to meet energy gap. Consequently, the energy density of complementary foods should be greater than that of breast milk. The quantity of complementary foods should be gradually increased every month while continuing to breastfeed. The consistency of complementary foods should also be gradually changed based on the child's age and neuromuscular development. Thicker or more solid foods provide more energy and nutrient density compared to thin, watery, or soft foods. Feeding thicker and solid foods helps to easily introduce several nutrient-rich ingredients, such as animal source foods. The frequency of meals should be based on the amount of energy needed to cover the energy gap, energy density of the food, and amount of food a child can eat in one meal. Meal frequency should be increased for children with higher energy requirements, or if serving lower energy density foods. Caloric needs will vary for children of the same age. The amount of nutrients recommended for each age group is referred to as dietary reference intake in the United States of America and as recommended nutrient intake in most of the other countries in the world. The following are general guidelines and will not be appropriate for all children. Growth should be monitored using growth charts to ensure a child's growth is appropriate, and feeding should be modified as needed. A 6 to 8 month old child needs approximately 2 to 3 meals a day. Initially, feed 2 to 3 tablespoons and gradually increase to half of a 250 milliliter cup per feed. A 9 to 11 month old child needs approximately 3 to 4 meals, each feed being half of a 250 milliliter cup. A 12 to 23 month old child needs approximately 3 to 4 meals, each feed being 3/4 of a 250 milliliter cup. Offer 1 or 2 nutritious snacks, depending on the child's appetite. In order to provide all the nutrient needs of a growing infant, add a variety of food sources to staple foods used for complementary feeding. Include dairy products, pulses, orange-colored fruits and vegetables, fats and oils, and animal source foods. Ensure that complementary foods provide sufficient quantities of micronutrients such as iron, vitamin A, zinc, calcium, and vitamin C, which are essential for growth, development, and prevention of illness. Based on local food availability and cultural practices, complementary foods can be offered as a thick porridge made of maize, cassava, or millet, along with milk, soy, ground nuts, or sugar added. Snacks may include egg, bread, yogurt, fruits or milk, puddings made of milk, biscuits or crackers, and bread with honey or bean cakes. During illness, infants need more fluid, so offer more fluids and continue breastfeeding on demand. However, the child's appetite for food usually reduces. Therefore, give small amounts of food more frequently. Once the child recovers and appetite improves, offer extra food at each meal or snack every day. The WHO recommends daily intake of an unfortified diet comprising a minimum quantity of the typical staple food, legumes, egg, fish, or chicken, and green leafy vegetables. However, such a diet has also been reported to be associated with micronutrient gaps, especially iron and zinc. This diet meets only 26% to 37% of iron needs, and 68% to 82% of zinc needs for children aged 6 to 8 months, and 35% to 52% of iron needs in children aged 9 to 11 months. However, such a diet provides 83% to 100% of zinc in children aged 9 to 11 months. Feeding a child breast milk frequently, a morning meal of cereal porridge, a midday meal of rice, beans, and orange, and an evening meal of rice, fish, and green leaves, plus snacks containing banana and bread with margarine, still leaves an iron gap. Providing fortified staple foods may reduce such a gap. Now let us discuss the desired behaviors to be followed during infant and young child feeding. Parents should be instructed to follow appropriate bottle feeding practices. Bottle propping should be avoided. Only breast milk or formula should be fed through the bottle, unless otherwise specified by the physician. Parents should be instructed to observe for satiety cues and not overfeed the child. Bottles should be used with formula until the child starts to feed with sip cup. By 9 months, the child should be introduced to foods through a cup. At 9 months, there is usually a reduction in the consumption of fruits and vegetables, and an increase in the consumption of non-nutritive finger and snack food. Therefore, parents should be instructed to offer 2 to 3 healthy and nutritious snacks to their child every day after 9 months of age. Juice should not be introduced until the child is 6 to 9 months old. Children should not be introduced to sugar-sweetened beverages. Feeding unfortified complementary foods that are mainly plant-based may not be sufficient during the complementary feeding period, as unfortified foods do not provide iron, zinc, and vitamin B6 in sufficient quantities to meet the recommended nutrient intake for children aged 6 to 23 months. Addition of animal source foods may help to meet the gap to a certain extent. However, the amount of animal source foods offered to children is usually not sufficient to meet the iron gap. Hence, include iron-fortified complementary foods, foods fortified with multi-nutrient powders, or use vitamin-mineral supplements. Home-based fortification strategies, such as the use of fortified sprinklers and fortified complementary foods, effectively deliver iron and zinc to children aged 6 to 24 months. Fortified sprinklers and complementary foods are easy to deliver and mix, and do not alter the appearance, taste, and organoleptic properties of the food. They also have long shelf life, are inexpensive, have very low risk of overdose, and can easily provide the required quantities of iron and zinc to the child. According to the World Health Organization's Department of Nutrition for Health and Development, children who are well-nourished during the first 2 years are likely to stay well-nourished for the rest of their childhood. According to insights provided by the HAB-EAT study on effective strategies to improve complementary feeding practices, introducing a variety of vegetables to infants can increase acceptance of new vegetables. Repeated exposure to the same vegetables can increase the child's acceptance of that vegetable. Familiarity with different vegetables is likely to increase consumption of a variety of vegetables and fruits as the child grows. Regardless of the reaction expressed by the child on the first trial of a food should be offered at least 8 times before deciding that the child does not like it. In conclusion, complementary feeding period is the crucial window of opportunity to prevent undernutrition and its long-term negative outcomes. Beyond 6 months of age, breast milk is insufficient to meet the increasing energy and nutrient demands of the growing infant. Currently, complementary feeding practices can be assessed at individual level using the Feeding History Job Aid or Feeding Practices Monitoring Tool. Evidence and expert-based guiding principles of complementary feeding are available to support the health professionals. Food fortification is important to ensure adequate intake of micronutrients. Complementary feeding period is also important to set the stage for establishing future healthy eating habits. Welcome to PGPN. We will discuss the emerging importance of gut microbiota in pediatrics. Gut microbiota, or the intestinal microbial milieu, represents a complex, dynamic, and diverse ecosystem in humans. Typically, about 100 trillion bacteria, comprising hundreds of species, are seen in the human gut. The human stomach contains very few microorganisms, and as you move down to the intestine, the number of bacteria progressively increase. The large intestine remains heavily populated by anaerobes, which constitute about 10 to the power of 12 cells per gram of luminal contents. Each individual possesses a distinctive pattern of bacterial composition that is based on the host genotype and initial colonization at birth via vertical transmission. Immediately after birth, the gastrointestinal system of a newborn gets transformed into a complex ecosystem. The majority of healthy, full-term breastfeeding neonates are colonized by heterogeneous bacterial microbiota within 7 to 10 days of age. Facultative organisms, such as gram-positive cocci, enterobacteria, and lactobacilli, are the initial colonizers of the infant's gut. Contact with microbes begins in utero and proceeds in a stepwise manner during birth and early infancy. Earlier, it was believed that the womb was sterile. However, emerging evidences indicate that bacteria are found in the umbilical cord amniotic fluid, meconium of healthy term infants, and fetal membranes. The meconium membranes predominantly contain Enterobacteriaceae and lactic acid bacteria. The other sources of maternal microbial transmission include the oral cavity, the mammary glands, and the sebaceous skin surrounding the breast. The mode of delivery also plays a role in the onset and modulation of the newborn's gut microbiota. Vaginally born infants acquire bacterial communities that are similar to their mother's vaginal microbiota. Cesarean-delivered infants acquire bacterial communities that are similar to the skin surface communities of the mother. The feeding method, along with the mode of delivery, significantly impacts an infant's intestinal microbial milieu. Maternal breast milk contains live culturable bacteria. Breastfeeding has been linked to the colonization of infants' gut by Bifidobacterial and Lactobacillus species, among others, and high assimilation of oligosaccharides, which is a substrate for these bacterial species. Breastfeeding is also linked to protection against pathogenic bacteria, as these oligosaccharides compete with their target ligands. Formula feeding, on the other hand, is linked to gut colonization by Bacteroides and Clostridium cacoides and low assimilation of oligosaccharides. Colonization with Bacteroides decreases the chances of Clostridium difficile pathogenicity. Therefore, decreased Bacteroides and Bifidobacterium populations in infant gut due to the use of antibiotics may increase the risk of Clostridium difficile infection. The introduction of complementary feeding has a significant impact on the microbial ecosystem of breastfed infants. Once solid foods are introduced, the bacteriological profile becomes more and more similar to that of formula-fed infants. A significant increase in the count of enterococci and enterobacteria and the appearance of bacteroids, clostridia, and other anaerobic streptococci is observed. By 1 year of age, infants exhibit a distinct microbial profile. By 2 to 3 years of age, the microbiota composition resembles that of an adult. Early infancy provides a window of opportunity for modulating child's growth and development via dietary interventions. Gut microflora confers multiple beneficial effects to the host, and these include protective, metabolic, and trophic functions. Protective effects include supporting host immunity and defenses. Metabolic function includes supporting digestion. Trophic function is related to crosstalk with the immune system and influences cell growth and differentiation. Gut microbiota and the host have a mutually advantageous interaction in conferring immunity. These interactions include mutualistic, parasitic, and commensal. Commensals play an important role in functioning of the immune system, and they compete for nutrients and produce antimicrobial peptides and metabolites. The immune system and commensal microbiota are sensitive to change in diet. Next, we will discuss the mechanisms by which these gut microbiota confer immunity. There are multiple mechanisms by which gut microbiota offers protection against infections. The gut microbiota prevents exogenous pathogen infringements by activating the immune system, as well as directly interacting with pathogenic organisms. Gut microbiota also has the ability to secrete antimicrobial substances, such as bacteriocins, that inhibit the proliferation of other bacteria. The activation of immune system and other functional modulations, such as electrolyte and water absorption and hormonal secretion, have been linked to the production of short-chain fatty acids by the fermentation of non-digestible carbohydrates by gut microbiota. Dysbiosis between the host and gut microbiota may be associated with several intestinal and extraintestinal disorders. These include immune-related disorders, such as atopy, and allergy and intestinal disorders. This indicates that there may be several factors that influence the gut microbiota soon after birth, which may lead to potential harmful effects later in life. Hygiene hypothesis implies that lack of microbial exposure or colonization in early life may increase the risk of development of atopic disease through insufficient stimulation of Th1 cells. A new hygiene hypothesis, or a Western lifestyle hypothesis, proposes that Th1 cells are not stimulated to balance the Th2 cells, but the imbalance in the Th2 cell population leads to the production of interleukins, which induces the production IgE and eosinophils, causing atopic diseases. Studies have shown lower rates of allergic sensitization and asthma in childhood in infants exposed to household pets or farm animals early in life. Biodiversity of the body's microbiota has been negatively associated with the risk of developing asthma and allergy. Disturbances in the gut microbiota have been shown to affect gut motor function and cause gas production, which results in abdominal pain and colicky behavior in infants. The findings of a comprehensive analysis of the fecal microbiota from infants suggested that microbiota composition in infants with colic differed considerably from the healthy infants. Lower counts of lactobacilli and bifidobacterium and higher numbers of gram-negative bacteria, such as proteobacteria, included Escherichia and Klebsiella, were observed in the stools of infants and colic compared to the control population. Given the small sample size of the study, further research is needed to better understand the relationship between colic and gut microbiota composition. Let's look at the importance of gut microbiota in diarrheal diseases and gastroenteritis. The most common etiologic agents of diarrhea in infants include rotaviruses and bacteria such as Campylobacter and Salmonella. Changes in the secretion and absorption of water and electrolytes via intestinal mucosa form the foundation of gastroenteritis and diarrhea. In this context, modulation of the gut microbiota may be a useful supplemental strategy for helping to control the symptoms and complications of diarrhea. However, further studies are needed to assess the benefits. Mounting evidence on the role of the gut microbiota in diseases has led to several research trials evaluating the manipulation of gut microbiota as a useful strategy to achieve microbiota balance. Evidence on the benefits of gut microflora manipulation are equivocal, and it is not currently a recognized treatment in pediatrics. The general approaches to manipulating the gut microflora include use of prebiotics, antibiotics, probiotics, and symbiotics. Prebiotics promote the growth of commensal bacteria especially, and examples include fructooligosaccharides, bran, and inulin. Probiotics refer to exogenous supplementation of intestinal and colonic microbiota with beneficial species, and examples include Bifidobacteria and Lactobacilli. Symbiotics refers to exogenous supplementation of intestinal and colonic microbiota combined with prebiotics. Immunologic benefits of probiotics include activation of local macrophages to increase antigen presentation to B lymphocytes and increase secretory immunoglobulin A production, both locally and systemically, modulation of cytokine profiles, induction of hyporesponsiveness to food antigens. Non-immunologic benefits of probiotics include digestion of food and competition for nutrients with pathogens, alteration of local pH to create an unfavorable local environment for pathogens, production of bacteriocins to inhibit pathogens, scavenging of superoxide radicals, stimulation of epithelial mucin production, enhancement of intestinal barrier function, competition for adhesion with pathogens, modification of pathogen-derived toxins. Prebiotics may confer benefits due to their metabolic effects by the 1. Production of short-chain fatty acids, 2. Involvement in fat metabolism, 3. Absorption of ions such as calcium, iron, and magnesium. They also have a role in enhancing host immunity by their involvement in immunoglobulin A production and cytokine modulation. A great deal of research is ongoing with respect to probiotics use in the prevention and treatment of digestive disorders, such as diarrhea caused by infections, antibiotic-associated diarrhea, irritable bowel syndrome, and prevention of necrotizing enterocolitis in very low birth weight infants. Some specific strains of probiotics have been found to be promising for infant colic and irritable bowel syndrome, but less is known about childhood constipation. The association between the early exposure of probiotics and islet autoimmunity was evaluated in the environmental determinants of diabetes in the Young study among 8,676 children at increased genetic risk of type 1 diabetes mellitus. The study findings suggested that in children with the highest genetic risk of type 1 diabetes mellitus, early probiotic supplementation may confer a lower risk of islet autoimmunity. It should be kept in mind that not all probiotics are the same, and the clinical benefits of probiotics are strain-specific or depend on the mixture used. The Agency for Healthcare Research and Quality states that reporting of adverse effects with probiotics needs to be standardized in order to obtain clearer conclusions about their safety. The NIH states that certain population groups may be at risk of side effects with probiotics, and these include, among others, critically ill patients, those who have had surgery, very sick infants, people with weakened immune systems. In conclusion, the colonization of the gut microbiota begins in utero. Development of gut microbiota in early infancy is influenced by prenatal, perinatal, and postnatal factors. By 2 to 3 years of age, the composition and diversity of gut microbiota of an infant resembles that of adults. Gut microbiota and the host have a mutually advantageous interaction in conferring immunity. Dysbiosis between the host and gut microbiota has been implicated in causing several intestinal, metabolic, and extraintestinal disorders. Modulation of the gut microbiota may have a role in childhood diseases. The clinical benefits of probiotics are strain-specific or specific to the mixture used, composition, biological activity, and dose. Therefore, the risks and benefits should be carefully considered before a patient decides to use them, as they are not regulated and available over the counter. The NIH have identified populations that are at increased risk of severe side effects with probiotics. The Agency for Healthcare Research and Quality has recommended the need to standardize the reporting of adverse events with probiotics.